The non-self. Document 3. Pathogens. Pathogen mean patho, disease, gene generator. Pathogenic agents are any agents that enters our body and cause inflammation and the formation of diseases. There are many types of pathogens. They can be classified into 3 categories. Viruses of size 10 to power minus 8 meter. They are non-living organisms made up of a DNA or RNA covered by a capsid protein. Why they are non-living things? Since they can multiply or remains alive unless they are present inside a target cell. Target cell like a human cell or animal cell, in which it integrates the DNA or RNA in the nucleic acid of the host cell, that is target cell, and multiply. Okay, for that the viruses are non-living organisms. Example of viruses like influenza virus causing flu, human immunodeficiency virus causing AIDS, and many others present on the table, document A. Bacteria, their size is 10 to power minus 6. They are also microorganisms, but living one. Example, streptococci causing pneumonia, otitis, and meningitis. التهاب الرئة pneumonia, otitis التهاب الأذن, meningitis سحاي. Okay, salmonella, typhoid, etc. We only want 2 examples from each. Fungi, protozoa, and worms. Their size is between 10 to power minus 2 to 1 meter. They are macroscopic. Sometimes they can be seen by naked eyes. Example, Candida, causing mouth lesion. They are fungi. Plasmodium, causing malaria. They are of protozoa class. And Ascaris and Tania, which cause intestinal infections. Okay? Okay. Antigen. What is antigen? As a definition, antigen in a simple way, we can say that antigen is any substance that enters our body and cause immune response. Okay? These antigens can be in simple form, simple molecules such as proteins, simple proteins, carbohydrates, or complex like polysaccharides, a junction between protein and lipid, lipoprotein, junction between protein and carbohydrate, complex one. Or an antigen may be foreign substance from the environment like chemicals, bacteria, viruses, or pollen. That can be soluble in blood. An antigen may also be soluble molecules but fixed on the membrane of tissues. Elements of non-self. Non-self antigens can be complex molecules such as proteins, carbohydrates, or living organisms or cells such as red blood cells. Complex molecules, protein, carbohydrates, they may enter our body and cause diseases, okay? Like enzymes. Taking an enzyme, taking a hormone, this may cause a disease. Cells, example, red blood cells, like blood transfusion. During blood transfusion, If the blood group is not compatible, this may, as we know, this may cause intravascular lysis and high fever. Okay, soluble antigens, example, toxins, venom. Do you know what do we mean by toxins? Toxins are harmful substances that enters our body. Chemicals. Venom is a solution secreted by snake and modified self cells. Additionally, non-self antigens, we can add toxins, as we mentioned above, those that are attached to certain tissues. Okay, once chemicals enters our body, it may bind to certain cell membranes or tissues, and causes diseases. Modified cells are cells that was normal, and under genetic mutations, these cells carrying self-HLA self-peptide will cause modification and lead to the transformation into a modified cell carrying self-HLA and synthesizing non-self protein like cancer cell. All cells of the body is recognized by the immune system as self by giving the identity card. All the polypeptide chains of HLA will represent self-HLA and self-peptide. Once a mutation is done or giving variation in the genetic information, this will cause expression of non-self protein, which are represented on the membrane of the cell. So the self cell will manufacture non-self peptide and represent it on the membrane by the polypeptide chain of HLA. Then the self cell will become modified cell carrying self HLA and non-self protein. Okay? Cells of the immune system, document 4. The blood is made up of many types of cells and molecules, mainly red blood cells. Red blood cells, we observed them previously. They are made up of only cell membrane and red pigment called hemoglobin, on which antigen are present as self-markers on the membrane of red blood cells. Today we want to see white blood cells, the immune cells. In order to observe them, we should mention previously that the blood is made up of three components, main components, or four, sorry. Red blood cells, white blood cells, plasma, and the platelets. All blood cells are originated from the bone marrow. These cells differentiate as they get mature to give different components and parts of the blood. The procedure for the observation of a blood smear, we should apply the following. At first, prick the tip of the finger with a sterilized lancet. Place a drop of blood 1 centimeter from end of the slide. Then we should decline it. Put another slide beside the drop of blood. Push the upper slide rapidly toward the opposite end of the lower slide to form a thin layer of blood. Allow the smear to dry. Add a few drops of alcohol of 95 degrees to fix the smear. Wait for 2-3 minutes, then add few drops of using for coloration. Wait for 7 to 8 minutes, then rinse with water. that is immerse the slide in the water. Add a few drops of methylene blue, wait for 3 to 4 minutes, then rinse the preparation with water and let it dry. And lastly, place the preparation on the stage of the microscope and observe it. As it represented here, here is some human red blood cells, red activated platelets colored purple. There is some monocytes as shown in green color, T lymphocytes, but a kind of white blood cells colored orange. This is some blood sample, okay? From it we have only to observe the white blood cells, which is colored violet, colored green and orange, okay? Immune cells are all originated from stem cells called pluripotent stem cells. These pluripotent stem cells are responsible for the secretion of different cells in our body. Pluripotent stem cells can be classified into two major groups, myeloid stem cells and lymphoid stem cells. Myeloid stem cells can be classified into three groups: granulocytes, mastocytes, and monocytes. Granulocytes can be classified into three cells called eosinophil, basophil, and neutrophils. Mastocytes and monocytes are the second and third types of cells corresponding to myeloid stem cells. Pluripotent stem cells also give lymphoid stem cells. Lymphoid stem cells can be classified into two major groups, T lymphocytes and B lymphocytes. In the coming slides, we will talk about each cell and their characteristics.